Hi, I'm Jacob, and I'm going to tell you about the cell of a bacteria. So the structure, it starts on the outside. We have the capsule, which you can see in red. It's the outside of the cell. It's the outermost layer. Some may have this slime layer made of polysaccharide, which enables organisms to attach to surfaces. It protects it from dehydration and attack by phagocytic cells and makes pathogens more resistant to our immune responses. Then we have the tail. It's called the flagellum. As you can see, some bacteria cells have it, some don't. Plural, you call them flagella. These are used for locomotion, so the tail will whip like propellers to get the bacteria around. Then we have these little pili, these little hair-like things. These are used for attachment to surfaces of other cells, help it move around. The sex pili is responsible for DNA exchange. A sex pili holds two bacterial cells together and allows DNA to be transferred between them in a process called conjugation, bridging the cells. Prokaryotes also reproduce asexually and usually takes place by binary fission. Reproduction can be very rapid. Rapid evolution of prokaryotes means they are proactively changing. Next, we have the cell wall, which you can see here is in blue. It's between the yellow and the red. The cell wall is a protective layer responsible for the organism's shape, prevents the cell from bursting when it takes up water. The cell wall of most bacteria contains peptidoglycan, a polymer of linked sugars and polypeptides, L amino acids, the type normally used to make proteins, and D amino acids, mirror images of the L amino acids. The cell wall allows them to survive in both hypertonic and hypotonic aqueous conditions. Next, we have the plasma membrane that you can see in the yellow. This functions as a barrier for the cell and separates the cell from its environment. The basic building block of the plasma membrane is the phospholipid, a lipid composed of glycerol molecule attached a hydrophilic water-attracting phosphate head and two hydrophobic water-repelling fatty acid tails. Next, we have the cytoplasm. That's going to be in white, just past the yellow, everything you see. This is the complex solution of organic molecules and salts inside the cell. Everything found inside the cell membrane, thus, it is the medium for chemical reaction. All of the functions for cell expansion, growth, and replication are carried out here. We also have the ribosomes. They're inside the cytoplasm. They're the little green circled spheres that you see here. This is where protein synthesis takes place. They link amino acids together very fast. Then we have the nucleoid region in purple here in the middle. This is the area of the cell holding the circular double-stranded DNA genome. The chromosome, usually single, is found here. This region lies within the cytoplasm, since these cells lack a membrane-bound nucleus. Lastly, we have the plasmid. Here's a little blue ring. It's present in the cytoplasm area, separate from the purple that you see. This is an extra chromosomal DNA, physically separate from chromosomal DNA. Most commonly found as a small circular double-stranded DNA molecules. Thus can be used to transmit relevant traits, which is beneficial to the genes to survival. Some beneficial genes to the survival. Okay, that was the structure of the bacterial cell. Now we're going to talk about bacteria. Bacteria is prokaryotic. The domain archaea constitutes the rest of the prokaryotes. Prokaryotes are unicellular organisms that lack membrane-bound organelles and other internal membrane-bound structures. They come in various shapes, but may fall into three categories: the cocci, which are spherical, bacilli, which are rod-shaped or rectangular rounded, and spirally, which are spiral-shaped. We have Koch's postulates identify the organisms responsible for specific diseases. An organism can be identified as the cause of disease when it is present in all infected samples and absent in all healthy samples, and it is able to reproduce the infection after being cultured many times. Interestingly enough, over 99% of bacteria and archaea are actually unculturable, meaning it cannot be lab grown. Prokaryotes were the first inhabitants on Earth. These organisms are abundant and ubiquitous, meaning that they are present everywhere. The first organisms were prokaryotes that must have withstood the harsh conditions of an early Earth. There's also microbial mats, which are large biofilms or microbial communities, which may represent the earliest forms of prokaryotic life on Earth. They grow mostly on moist surfaces. As you can see, here is my oceanic setting. A microbial mat is a multi-layered sheet of prokaryotes, mostly bacteria, but also archaeans. Some prokaryotes would gain their nutrients from the chemicals in hydrothermal vents, while others began using sunlight once photosynthesis evolved. Some soil bacteria are able to be formed on spores that resist heat and drought, thereby allowing the organism to survive until favorable conditions recur. These adaptations allow bacteria to remain the most abundant life form in all terrestrial and aquatic ecosystems. The Dead Sea is hypersaline, but salt-tolerant bacteria thrive in this sea. To live, prokaryotes need a source of energy, a source of carbon, and some additional nutrients, macronutrients from the abbreviation CHOMPS, and micronutrients, both macro and micro. Bacteria and archaea that are adapted to grow under extreme conditions are called extremophiles, meaning lovers of extremes. Now I'm going to talk to you about the three so-called Terrabacterias. First is cyanobacteria. Cyanobacteria is one of the most common organisms on the planet. They have the capability of using sunlight and carbon dioxide to survive exclusively on those nutrients, which they turn into energy. They have remained almost unchanged for 3.5 billion years and were the first organisms to evolve in the process of photosynthesis. Today's usage of cells functions includes harvesting fuel cells, or fuel, which is ethanol, from genetically modified cyanobacteria, trillions of them, into a biofuel production plant by a company called Juul Unlimited. So this is one of the applications of cyanobacteria. Which you can see here. I'm going to talk about a surveying study that was done in Antarctica regarding cyanobacteria. This is one of the extreme environments in which this type of bacteria cell survives and actually thrives. Antarctica is inhabited by microorganisms that have adapted themselves to extreme environments and rely on symbiotic associations such as biofilms, like these. Antarctica is possibly the best studied region with respect to diversity, distribution, and cell biology of these microorganisms. As you can see here, this is one of the biofilms with the cyanobacteria. We have that these microbial mats are of a conical shape, and they have this maroon top layer produced by the cyanobacteria. The names of these are Oscillatoria sp and Formidium sp, which I tried to replicate in my model here. Conical shaped with the maroon on top. It was found that cyanobacteria in Antarctic lake ice water pockets, where the temperature is always under zero, were metabolically active and retained their photosynthetic cap, shown to metabolize even at negative 20 degrees Celsius, meaning they are still chemically reacting and generating energy, thus reproducing. Cyanobacteria are the only known prokaryotes able of oxygenic photosynthesis. They employ various strategies that compensate for osmotic and the mechanical shock that is created by freezing. Cycro-tolerant bacteria are capable of synthesizing extracellular ice-bending proteins as well as antifreeze proteins, which reduce the water freezing temperature. They can also protect their DNA from UV radiation. Antarctic microbial communities are in fact the lost world because they have been subject to isolation with a relatively low disturbance level. Cyanobacteria were shown to dominate in the Antarctic microbial mat surface layers, so they're mostly found on top, while deeper layers were inhabited by phototrophic bacteria of the phyla Chlorobi and Chloroflexi. Next, I'm going to go into how cyanobacteria can be very toxic, can become a toxin in water, as you can see here. Basically looking like pond water. There's like plankton inside the water, and it's just riddled with bacteria. Instead of blue, we have this green hue that I also tried to replicate here in my model. Here is we have something like a pond, where it's just like green on top, green inside, just bacteria everywhere. Cyanobacteria are ecologically one of the most prolific groups of photosynthetic prokaryotes in marine and freshwater habitats. They are primary producer microorganisms and are involved in the production of important secondary metabolites, including toxic compounds, such as these cyanotoxins. Environmental conditions, like climate change, promote massive growth of these microbes, causing blooms that have critical ecological and public health implications. These are the blooms. Just basically is an uprising and increased level of the bacteria. In certain water bodies, they can form dense blooms. In certain environmental situations, like when eutrophication, carbon dioxide concentration, water temperature, and/or high light intensity increase. Blooms can cause water quality problems, increase turbidity, and smother submerged aquatic vegetation, killing the vegetation. Oxygen depletion also kills the fish. Odor and taste is found in waters. The production of the toxins that can cause pathologies when ingested by birds, mammals, or humans. Now I'm going to talk about the Deinococcus radiodurans. They are well known for its extreme resistance to radiation. Desiccation and various DNA damaging chemicals such as mitomycin C and hydrogen peroxide. Data suggests that radio resistance of Deinococcus radiodurans is a combination of multiple strategies, including protection of proteins against oxidation, efficient DNA repair pathways, a condensed nucleate structure, favoring the maintenance of DNA fragment cohesion after irradiation. Global analysis of the Deinococcus radiodurans genome expression allowed the identification of a series of genes whose expression is induced after irradiation or desiccation. Recently, they have shown that DDRC is a DNA binding protein that binds single and double stranded DNA with a preference for ssDNA, protects DNA from nuclease attack, and exhibits a DNA strand annealing activity. A new study by Cornell Research reveals that nitric oxide, a gas molecule used in many metabolic processes in animals and a pollutant in the atmosphere that leads to smog, plays a key role in Dana-Cox's recovery when exposed to ultraviolet radiation. Lastly, I'm going to talk about the actinobacteria, the last of the Terrabacteria, which you can see here, and also replicated a small model, as you can see here in red. Actinobacteria, isolated from natural heavy metal ecosystems, may be a valuable source of novel secondary metabolites and therefore of new biotechnologically promising antimicrobial compounds. Actinobacteria are a group of gram-positive filamentous bacteria with high G and C DNA content and are ubiquitously found in aquatic and terrestrial environments. These bacteria represent the most renowned group of microorganisms for the production of bioactive compounds with diversified chemical structures. They are well known for antibiotics and anti-tumor agents production and are considered the source of about 45% of the obtained molecules from the microbial origin. Among actinobacteria, Streptomyces is still considered the best important source of bioactive secondary metabolites. Generally, 80% of discovered antibiotics are produced by the genus Streptomyces, and rare actinobacteria, such as Actinomedura, only 20% are produced by fungal species. Investigating actinobacteria in unexplored habitats and other extreme environments in which certain physical and/or chemical factors differ significantly has shown great importance because of the novelty of these antimicrobial compounds. Morocco has a distinctive climate and geographical position, with saline biotopes, arid deserts, thermophilic sources, and polymetallic mines, providing researchers with much to explore. The genus Actinopolymorph was for the first time isolated from a mining ecosystem. This is according to a study I read in Actinobacteria in Morocco. And that's it. That was so long, huh?